Dr. Tour, thank you very much for being here, sir. And this is really exciting for me to have you. And please let me first introduce you. So to the audience, first, Salaam Alaikum, peace be upon you all. And today we have a very special guest on Finding Truth, the renowned Dr. James Tour, who is not only a scientist, but also a believer. So and he always starts his sessions with mentioning that, which is really spectacular in today's world. So welcome, Dr. Tour, to this interview. And really, thank you very much for being here.
Well, thank you for having me, Ahmed. It's really my pleasure. I've always had a warm welcome from friends when I've gone to the Middle East, and it's interesting in so many ways. It's freeing because I'm able to talk both about science and my love for God with Muslims much more than I can with people in the West. So when I go to the Middle East, it's in many ways it's refreshing for me because people are far more open to speaking about God.
Yeah, it's a blessing that there is some parts of the world where you can still do that, isn't it?
Yeah, so, we have the freedom to do it. It's just that when you do it in the West, sometimes people think you're crazy.
Yeah. Oh, no. So first I have to introduce you, and I have... it's a difficult task because really your CV is huge. So I'll just give a glimpse about it. For those who don't know Dr. James Tour already, James, you're a renowned synthetic chemist. You've been named the Scientist of the Year by the R&D magazine in 2013. You have been ranked as one of the top 10 chemists in the world over the past decade by Thomson Reuters. And I was personally fascinated when you went public with your scientific criticism to origins of life research and the media ramp up of the real status quo of what we know about origins of life. For those who don't know, Dr. Tour has recently given a full course about abiogenesis on his channel. And his channel is Dr. James Tour. That is Dr. James Tour, YouTube/C/Dr. James Tour. For those who are interested to look at the course, I have actually watched every minute of it, sometimes more than once. And it's like 13 episodes, and I guess there's one of them has 2 parts or something. And Dr. Tour is TT and WF Chow, professor of chemistry, professor of computer science also, which makes him very close to my heart because I studied electronics engineering and communications and computer science and control. He's also a professor of material sciences and nanoengineering, so quite a mix, in Rice University, in the Smalley Pearl Institute, and the Nanocarbon Center. And of course, his work with graphene is cutting edge. Something about his career. So Dr. Tour is primarily focused on carbon materials, chemistry, and nanotechnology. He has over 700 research publications. And since my family is in the research field, I know what 700 research publications means. So way to go, Dr. Tour, of that. And 140 patent families. The last number I had was 130, and he updated me. It's 140 now. So it's very clear he's really doing more every day. And his H index is 156, which is a big deal. And his citations, which is the number of times other researchers refer to his work, to his literature, is over 110,000. And this is a very big number. In terms of awards, he became a fellow of the Royal Society of Chemistry in 2020, was awarded the Royal Society of Chemistry's Centenary Prize for Innovations in Materials Chemistry with applications in medicine and nanotechnology. So Dr. Tour is helping us in both our health and creating those small nanobots that maybe someday would unclog an artery or something. And he was inducted into the National Academy of Inventors in 2015, named among the 50 most influential scientists in the world today by the bestschools.org in 2014, named Scientist of the Year by the R&D magazine in 2013, won the ACS Nano Lectureship Award from the American Chemical Society in 2012. He was ranked one of the top 10 chemists that we already said by Thomson Reuters in 2009. And in the same year, was also made a fellow of the American Association for the Advancement of Science. And the CV goes on, so I'm going to stop here. Maybe I'm going to refer to his work on nanocars later when we start asking him about molecular machines. But Dr. Tour, being wary of not finishing all our 90 minutes into your CV, this is one great CV. So congratulations, sir.
Thank you.
So our discussion with Dr. Tu today will have will be around abiogenesis and some aspects of evolution that relate to chemistry. And so let's first start by this. What drew my attention to this field specifically is that, Jim, the more I look into the mainstream, there is always this proposition that unguided chance events that are random can produce everything around us. And this, spans from how the cosmos was originated, random fluctuations and stuff, how genetic material came into being, primordial cells, how those cells then evolved into creatures, how we have humans, how we have conscious beings and awareness and intelligence. Everything, when there is no explanation, is chance. As a scientist who really goes into the lab and invents things and puts plenty of definite thought and work into that, is this a rational scientific position, according to your experience as a scientist and even an inventor?
No, it doesn't make any sense to me, really. I can't fathom this. We put so much work and so much detailed thought into trying to carry out even the simplest of chemical reactions, the new reactions that we do, to think that something like just even a living cell could have come about by a random process is just crazy hard. This is just silliness. And people will project this out and say, well, it got to a certain stage and evolution took over. And just by saying that, there's this this homage that they think has to be paid. Oh, evolution took over, and then we can take care of all of this. No, we can't just throw this at the doorstep of evolution and say evolution took care of this. And so this is an irrational approach. And I even see this in graduate students. You ask them, well, how did that happen? And they say evolutionarily. Well, because they've said evolutionarily, the audience now has to sit in quietness. Oh, okay, so evolution did it. Now we understand. No, I don't understand any of that, nor does anybody else. But because they use that word, we're supposed to somehow bow down and just accept it. And as soon as you ask for details, okay, take me through the process of how evolution would do that. Let's take it one step at a time. And then they get angry. Everything starts to wither around the edges, and you see that nobody really understands this. So just because we throw out that term that has become a sacrosanct term that you should not ever oppose it is a real problem, because it does an injustice also to our students to think that you can just throw out that word and now nobody can touch you. So we try to explain things, and this is not some holy word that's untouchable.
And actually, two things that you just said caught my attention. So the first one is being angry, which is something I experience every day, because due to the videos that I put out on my channel, I go into those conversations every day, tens of them. And the second thing is, do you see this in other fields of science? That this one word that is a banner would be given as an explanation for such multitude of things, and we have to accept it. So number one, what is your explanation for the anger? And do we see this in any other fields of science? Other real empirical fields of science?
Well, I mean, scientists are like anybody else, but I don't know. I don't know that there are fields of science where you see such anger as you do in this saying this word evolution and trying to not embrace it. We're seeing more of that around issues that are scientific and pseudoscientific, and things can move more toward the political, and then it gets like that very rapidly. But I think that's often normal human behavior, that if you don't have an answer, that you can react back with anger in a way to quench the argument and to stop it. And I think that's normal behavior, and scientists are just normal. I mean, we have our own fears. We want to allay our fears. We care about our reputation. We care about things that we've said and our work, and we don't like when people start publishing papers that contradict our work. I mean, it keeps us up at night. I mean, we're just like everybody else. So this is normal human behavior, I suppose.
So the takeaway from that is that scientists are not gods, are they?
No, they're not gods. And when we don't have answers, we often get angry. And you see this sort of thing also in religious arguments. When people stop having answers, they'll start getting angry, and they'll start shouting and pounding their fist on the table, as if that somehow has given them now authority to not have to answer certain questions. So when you don't have answers to questions, you have a tendency to react back with anger. If you know it very easily, you just answer it.
Yeah, that's what a reasonable person will do. So let's go to our second one. It's our opening question about life. And to me and to many others, life is an enigma to material science. I've never seen a clear explanation of what is life other than its material features. But whenever you go in a discussion, somebody will say it's just chemistry. So you're the chemist, and a renowned one. What is your reaction when people say life is chemistry?
This is bumping up against philosophy, and it's a hard question. So, for example, you could take a wonderful person, say Mother Teresa, who spent her life in India serving the poor, who had they take this oath of chastity, of poverty, and of service to the poor for the rest of their life. I mean, that's an amazing oath, that you're going to remain chastity, you're going to remain unmarried, you're going to remain in poverty, you're never going to seek money, and you're going to devote your life to serving the poor. So here is this great woman who did that. Now you could take a woman like that and you could give her 1 milligram. 1 milligram is like the size of a grain of salt. And you could give her 1 milligram of material, and that could affect her brain enough to make her into a mass killer, a mass murderer. So there's certainly something chemical going on there. So there's chemistry that happens that affects our brains, that affects our actions. On the other side of it, there's things that are very hard to chemically understand. It doesn't mean that because we don't understand it today, we will never understand it. It just means that today we don't understand it. So, for example, my granddaughter is visiting. I haven't seen her in almost 2 years, my two granddaughters. I mean, the younger one, she just stays by my side. She will sleep next to me. Wants me to hold her hand when she goes to sleep. I mean, she's right there. My love for her is so great. How do I explain that with science? How do I explain that with chemistry? I would sacrifice my life 100 times for her if I could. How do I explain that? How do I explain that when I'm working on all of these things and all of this great research and all of these great ideas, that 60% of my CPU is thinking about that girl? Why is that? What is it that makes me want to be with her and embrace her? These things are hard to understand. There is no doubt that within life, chemistry is exploited for the purposes of life. But is there something that goes beyond that? And certainly the materialist says, no, it's just the chemistry that goes on here. And there are other things, even within science, which gives hints that it extrapolates beyond the materialistic world. And I'm sorry for moving this conversation, but you see this, for example, in NDE, near-death experiences, where their heart has stopped, where they're and they are functionally dead for minutes, 5 minutes or something. And then they come back and they describe to you things that scientifically are very hard to understand, where they describe to you that they raised up out of their bodies and they saw things that were on the tops of high counters, high cabinets in the hospital room. And then they saw things on the roof of the building that they could never know were there. And then they go and they see these actual very features on the roof of the building. So there are things that happen even within science when you start probing these people as scientists that science is unable to understand with the materialistic world, and it has to start touching on the metaphysical. So even within science, we bump up against the metaphysical when it comes to talking about issues of life. So that's about as deep as I can go, Ahmed.
So is it fair that it is to say that it is unfair to life to just reduce it to chemistry with such a blunt statement?
It's certainly unfair in the respect that there's so much of the chemistry, even of the chemistry of life, that we don't understand, that we have no way today of explaining. Again, we might be able to explain this in 100 years or 200 years or 500 years from now. Just like if you asked a man in the 1700s, why is it that when the parents are tall, the child is tall? When the parents are short, the child is short. And they may say, well, this is God. Well, okay, well, this is God, but scientifically, how does this happen? They wouldn't know. Well, now we know that the genetic code is inscribed in the DNA, and the features, the enzymes action that's going to build the certain features is encoded from that master plan that's inherited from our parents. So we understand more today than we did 300 years ago about how this thing happened. So over time, we may not be able to understand it. It's unfair to say, well, this is just chemistry, because there's a lot of chemistry we don't understand. And it's unfair to say this is just chemistry because there are features that science does not explain for us, like the love of a man for his grandchild, or the love between a husband and their spouse, of the emptiness that's felt when one spouse dies after they've been married for 50 years, the emptiness that comes. How do you explain this chemically? Maybe there is a chemical explanation, but it's not fair to just say, well, that's just chemistry, any more than it's fair to say in this day and age, well, that's just, that's just inherited. Well, give me more details, or any more than it's fair to say, well, that's just evolution. Evolution does that. That's not fair. Tell me more. Give me more specificity.
Great. So it takes us to our next question, because while you're a chemist, in your final episode of your Origins of Life course, you posted what seemed to be a challenge. I think you called it a challenge, and it was about creating a living cell from scratch. And you even extended it more, and you said, "We'll give you the material, whatever material you want. We'll give it to you." So it's as if you take care of the chemistry, and then show us how from that you can create something. In my religion, for example, in Islam, there is a challenge that looks similar to that, where God challenges people who don't believe to create a fly. He says, "Bring everybody on the face of earth and create a fly." So let me ask you, what is the significance? Why did you choose to make that the closing thing, the closing challenge of your course?
Well, it's interesting with your Islamic challenge, because, you know, a fly is probably the smallest entity that the people could fathom in that day.
Yeah.
But a fly is exceedingly complex compared to a single cell.
Yeah.
And now 1,500 years later, we can't even make a cell, let alone a fly. I mean, the complexity in a fly is just fascinating, just fascinating. Even I have sat and I allow mosquitoes just to sting me, just for the joy of watching them. Because you watch this little mosquito flying, and there's this amazing coordination of flight. And their head is like the size of a pinhead, and tiny little brain, and has all this coordination, and it's able to detect that I'm viable for food. And after it stings, it draws the blood. Then immediately it emits something, another chemical that calls other mosquitoes and says, "Here's fresh food." You know,
He's a prey.
They call them. This is extraordinary scientifically. How do you do this? A fly. I mean, a fly is so hard to kill a fly. I mean, how do they react so quickly to be able to jump and then start flying so rapidly? So even,
you know, about the mosquito, we have another verse in the Quran that says, the translation of meaning is, Allah is not embarrassed to give an example or a parable with a mosquito and what is higher than it. So the ones who believe, they will know it is the truth from their Lord, but those who don't will say, "What is this example?" And it's amazing listening to you give the example of the mosquito. It's just amazing. Yeah, and you as a believer see what is the great wisdom behind it, and others would say, maybe it's chemistry or it's evolution or something.
Yeah, it's so hard to fathom these things. So just creating a single cell. So the challenge was, okay, you have to first make the building blocks of the building blocks. You have to first make the sugars. You have to first make the amino acids and the nucleotides and the lipids. So you have to first do this. And then I say, well, I know that's too hard because we don't know how to do this in a chiral fashion, and we don't know how to. But okay, I'll give that to you. Now you have to take them and polymerize them. Well, polymerizing them is very hard to do that. Polymerizing sugars is very hard, and it's not easy to do this. And so I say, okay, well, you can't make the sugars, and then how do you polymerize RNA and prevent the 2,5 linkage and only get the 3,5 linkage? Very hard to do that. How do you do that? Even if it's DNA, so it's deoxyribose, you still have to do the activation step in order to get these things polymerized. Without activation, it's very hard to get these sorts of structures, so they have to be activated. The chemistry is exceedingly involved. So I say, yeah, okay, I understand. It's hard to make the polymers. So I'll give you the polymers. Now you have to make the cell. So I'll give you the polymeric structures. Well, you have to have the polymers in the right order because the information is encoded in the sequence of the DNA and the RNA. You say, okay, we don't know how that happens. So I'll give you the sequence. You name the sequence, I'll give it to you just like you want. Now, if I gave you all those components, could you make a cell? And you don't have to do this in a cave someplace or in the bottom of the ocean. You do this in your pristine laboratory with all the reagents you want, all the literature you want. Can you do it? And the answer is no. The answer is no. And so nobody has picked up this challenge and say, yeah, I'm going to do this because we can't do it. So I took the simplest living system that you can have is a cell. And that is the simplest construct we have. In fact, that's how we define life. It has to be made of cells. That's not my definition. That's long been the biologist's definition. And it has to have homeostasis. It has to be able to have this metabolic functions. It has to be able to breathe. It has to be able to reproduce and have its offspring. And so it has to have all of these features to have life. And I went through and I showed all the different functions you have to have because biophysicists have reduced, theoretically, you can say, what are the functions you have to have for life? What are the functions you have to have? And you have to have so many different pieces that you have to have at least 200 protein coding genes. And even with that, you'd have to have all the exogenous amino acids coming in. Where do you get this? I mean, it's so hard to fathom. So no, nobody would pick up that challenge because as of today, it can't be done. Now, again, I can't say that it will never be done. I don't know where we will be in 500 or 1,000 years from now. All I know is that it can't be done today, and it is harder. It is going to be harder to think about how to do it next year than it is this year because the target's moving further away.
Actually, this is my exact next question. Okay. So it is, what do you mean when you say that the more time passes, the method of creating life becomes more difficult? Right. So, I mean, aren't there enough complexity that we have already discovered? Do you expect that there is much more complexity that we don't even know about to the extent that we're just at the crust of this?
Yeah, I mean, what happens is the more we learn about a cell, the harder it gets to think about making one. So 100 years ago, a cell was just a bunch of protoplasm in this cell. Well, now we know a cell is a factory. There's so much going on in there. And every year we learn more about the complexity of a cell. And each of these features that we're going to talk about today, I mean, it's hugely complex. So we will know more about the complexity of a cell 5 years from now than we know today. There are many things that we know about a cell now that we didn't know 5 years ago. That's the beautiful thing about research. You turn over one stone and you find 5 other mysteries. And so what happens is the target, the challenge to make a cell is here. And now in 5 years from now, it's out here. So the target gets further and further away. So we say the goalposts have been moved. So you know, in Egypt you play a lot of soccer, and you know, say you move the goalposts another 100 meters away, and it becomes harder to score a goal. And so you keep moving the goalposts further away faster than you're approaching it. So the goalpost is moving away faster than you're approaching it. That means that the target becomes harder. So actually, the target is further away each year because we go, whoa, I didn't know a cell was that complex. So long before we ever get to your challenge of a fly, we've got to make a single cell. And we have no idea how that's done. And then building the whole system of a fly, I mean, forget it.
In one discussion I was having with an evolutionary biologist, I just had it. So I said, okay, create a fly. And it was also with a science explainer, and I got this peculiar answer that I'll take your opinion about it before we go to abiogenesis. He said, well, nobody is picking the challenge of creating a fly because nobody cares to create a fly. And it just shocked me because I know that there are so many scientists and engineers working on trying to create bots like flies and mosquitoes. Is this the case, or I'm imagining it?
Well, if you could create a living cell, you'd win a Nobel Prize. So why somebody would say nobody cares about that? Just to make one cell, you'd win a Nobel Prize. I mean, like immediately. I mean, this is so big of a discovery. Now, to be able to make something as complex as a fly is just so unfathomably hard. What scientists do is they just think about this, and you have to understand. We make drones, and you can make little drones, and they can fly around. Well, what is that drone made out of? Made out of plastic. It's made out of composite. It's made out of silicon. Little patterned wires. Why don't you build that drone out of molecules? Why don't you build it out of peptides and nucleotides? Why don't you do that? Because we have ubiquitous all around us. We have the model of flies. And so we should copy that, because that's what people do. They see something and it's easiest just to copy what you have. So build Absolutely. It out of the very things that the fly is made out of. Nobody does that. Nobody does that because it's so unfathomably hard. What's been put before us in nature is the hard way to do it. God's put before us the hard way. We take the easy way by using all these composites and things, which in itself is not easy. I'm talking about hundreds of years of development to develop all the materials to be able to do this and the techniques to do this and the clean rooms and the silicon processing to make these little drones. Well, why don't we make it like a fly? Because those have far more maneuverability. Those run on far less energy. And those are able to acquire their energy on the fly, we could say, as they're doing things. And,
That's a good one.
And so why do we do this? Because we are so without knowledge on how to do this out of the raw chemicals that are there in nature. We don't know how to do it. So we try to mimic it with something else. So I don't know why anybody would say that, but I disagree with your friend that nobody cares to make a fly. Oh boy, that'd be just, I mean, you'd be so famous. You'd be so famous. You'd be on every talk show, everything, if you could do that. And you'd be very wealthy too.
I couldn't agree less. So what I think it's again about, these strange responses that one would get out of discussions with specific people who hang out to grab so much to the matter of evolution. So let's go into abiogenesis. How about that? So, and abiogenesis for the audience is the creation of life from non-life. Is this a fair description?
Yes.
Yeah. So my first question to you is like this. So science explainers, and those are the people who would go and give shows to people to tell them science in a simple way. They pretend that science has got life's origin figured out. And I guess your course was motivated by one specific science explainer who claimed that this is not a big deal and, talked about agendas and stuff. So my question to you, is that what one will find upon reviewing the real scientific literature? So if one grabs the papers and opens them, will he find that, the paper starts by saying this is a done deal and this is how it's happened? So is this what's there in the books, or it's an artifact of science explainers?
Well, certainly it's there in textbooks. But it's not there in the hardcore literature. And even the hardcore literature makes lots of unwarranted claims where they'll say they're just at the threshold. It's just a matter of evolution. It's just a matter of 1 million years of time from this point. But those are fallacious claims. Those are fallacious because you look for the details. It's not there. It's not there in the paper. And what happens is the uninitiated, the non-scientist reads those papers and they will pull that quote out and say, see, it says here in a peer-reviewed paper. Well, this is my argument. Even the peer-reviewed papers are full of trash. They're full of nonsensical comments. And so what happens is the people who write these papers, they never say these sort of things to me, because I know it's so easy for me to challenge them on that. They will say these sorts of things to the press, and the press will take this and run with it, or they will allow these sorts of things to go unchallenged into textbooks, such that so many people are misled. And you saw on that series, I talked about surveys that were done, which 80% of the people had college degrees of one type or another, between an associate's 2-year degree to PhD, and something like 70% of them, of the people, thought that scientists had made simple bacteria in a laboratory. Something like 60% of them thought that scientists had created simple life, like a frog. A frog is not simple life, but
it's more complex than a fly.
Right, but where did they get this? They got this from textbooks that say this sort of nonsense. So yes, this is pretend. This is not true, and so the world is highly deceived on this, unfortunately.
Actually, I had this one very, very long conversation, a written one, like 90 posts or something, with one very famous science explainer, and he actually went on his show later, and he put it in the open. And what he said, he told me this: I told you that textbooks, and he pointed to a specific university textbook, and it says that novelty comes from random mutation. So you cannot say no, because then you are talking against, a university textbook. So he said, if you're not agreeing with the textbook, then he's talking to me, then you have no credibility. So I tell him, so does anything that goes in a textbook has to be true? Is this the new definition of truth? And it's miserable, because when people who are not in, you're an authority, I can say, in your field, but when people who see that this does not make sense from another field like information theory or something, and talking to normal people, they would say, but it's in the textbook, and it really makes it hard. And so it's really miserable. So let me ask you this now. Even origins, now this is the bad one. So science explainers, maybe they are there for, making nice episodes or something, or a show. But even the origin scientists on the media act as if the matter is almost a done deal and that gaps are small. And I have it in mind, one of those guys who calls himself a professor. I went to his channel and I said, why are you saying this? And he just went ballistic and angry and started throwing things at me. So why is this a characteristic to the field of origins and evolution, in your opinion? I think we talked about that when we talked about anger. But I don't know. Is this... Scientists what should be the normal attitude of a scientist when something is not clear through science, in your opinion? I've seen you say we don't know so many times. Why don't they do it?
Scientists want to get... I don't know why people are pushing this as if they really know it. I can see how the non-scientists, like the people who say that they're professors and they have YouTube channels, I can see how they would not know. They just don't understand. They read something in a paper that says, the origins of chirality are solved. We have so many ways to get at this, and then they accept it, without looking at the details that this isn't solved at all. So if they just read the papers, they're deceived. So in some ways, they're the victim. They're a victim. They're victim of this type of writing in these papers. It really harkens back to the scientist. And so the responsibility is actually with them. This has to be deposited right at their feet. Why are they coming with this and saying that we've got this thing figured out, here's a way it happens, when they see the difficulties? And they understand what I'm talking about. They understand the difficulties of the things that I am putting forward. You see that no scientists have come out and taken that challenge that I put forward. You see that scientists are not coming out and dealing with the unprotected sites. I mean, these things are hard problems. Why they're not coming out and saying this? Well, if you're in the field of the origin of life, you have to continue to have funding for your research, and you have to continue to support that. So you're not going to say we don't know what's happening. You've got to say each year we're closer to this thing. Each year we're closer to this. But now I'm able to get them. And this is what I'm going to start to do, because they have said this and they've given time frames for when this thing will be solved. But because so many of their talks have now been recorded on the internet, I can start holding them to account.
Time frame. They have given time frames.
They've given time frames for
solving. And what kind of time frame did they give? This is new to me. Maybe I missed it.
Yeah, like a couple of years, 2 years.
2 years.
Yeah. We will have this. So
wow.
So that was 10 years ago. So now I'm able to go back and start calling this thing out there. And you see how this game is played. You keep saying we're close, we're solving this problem, and you keep getting support, you keep getting funding for it. Now, it's not like Origin of Life is a highly funded field. It's not. It is a boutique of a field. But they have a very influential voice in textbooks, in the way science is taught. It goes way beyond what the impact they should have for the science that they generate. It's really a small number of scientists working on this. So, it's nowhere near the funding that is given, for example, for making electronic devices or something. No, it's nothing to that scale. But even what they have a huge impact, though. And they have a huge impact, and you can see their impact based on the surveys that I had cited in there, that people are thoroughly confused on this thing. So really, all of this harkens back to them. And so they have to be held accountable for what they're saying. And that's going to start now being my target. I'm going to hold them accountable. I have their words. I have it recorded. And now we're going to start going after, because they've got to be careful what they say, because it's these YouTubers are, in a sense, the victims, because they take their words and they run with it. The poor press is clueless. These people in the press are absolutely clueless. They got their degrees in journalism, so it means that they're very good writers. But they don't know how to assess the science. So they hear scientists and they write articles. And very often, I know this because I talk to the press a lot, I would say 80% of the time, I am given the opportunity to vet the article before it appears. And I could say, no, you've extrapolated this too much. Let's back off on this. Because the press loves to ramp things up, because that's their business. If you can embellish something, that'd be great. It's up to the scientists to pull back on the reins and to say, slow down here. We really can't say this. We can't go in this direction.
Maybe they don't slow down. Maybe they let them just go, because I've seen some of them come into YouTube channels, and amazingly they say some very long shots.
But that will not go unchallenged.
Yeah. So it's music to my ears.
That will be the next target.
I'm really this is really music to my ears to hear that you'll be doing this, because once I had this other debate with a science explainer, and he said at the end, Ahmad, you're giving me opinion. I'm referring to papers. And all what I have is peer-reviewed papers. If this is what they say, this is what I say. And I say, okay.
But they don't have, the poor folks don't have the ability to assess the peer-reviewed paper. As you saw in my talk, in that I said, here is what they say, but here are the results from their paper. And I would go right back to the experimental sections, the supplementary information, and look at their own data and say, here's what their own data shows, how they made this extrapolation. So people will look at a peer-reviewed paper and grab one sentence without looking at the data itself, because the data is highly embellished. Now, look, as a scientist, I know how this game is played. I know how this is done. And so I realize, these things take place because you want to project your science. But now it's gotten way out of hand to where the poor man or woman on the street is thoroughly confused. thinking that scientists, oh, they've made cellular life. Even scientists have made simple things like frogs in a laboratory. They're so wrong on this that we got to pull back.
So it's great that you'll be calling them out now for this. Just for the audience, we'll be having a small recess, 5 minutes from now, and then we'll be having the second part. But let's try to touch on the RNA world hypothesis, which is this, claim that, since DNA is so complex, maybe things started as RNA, and then kaboom, you have coding for things, and you have primordial cells and this stuff. So let me ask you this question as a start. Now, I've been doing my work about looking into this RNA world hypothesis and LUCA and mutations and stuff, and I have been struck by the immense error rate when it comes to RNA replication versus DNA. So my question to you is it possible that there would be life based on RNA? I mean, a cell that does not have DNA at all, not a virus, a living cell that has RNA as its genetic material, given the huge RNA error rate compared to DNA. And I've, for example, went into literature and I saw that even after replication, a living cell, due to ultraviolet and other environmental conditions, will have its DNA damaged, and then it gets repaired by repair mechanisms. But there isn't the same thing for RNA. So there is no, up to what I have read, there is no RNA-based living cell today. So how come somebody would speculate that there were RNA-based living things at some point of time? As a chemist, do you think there can be a stable living cell that has no DNA whatsoever and only has RNA as its basic material, genetics? Is this possible? Did you see it?
I don't see it. Many people have suggested it. Other people, including many scientists, materialistic scientists, are saying that can't be. The problem with RNA, well, first of all, RNA is more complex than DNA is, in that each one of the bases is hooked up to a sugar that has one more stereogenic center than that in DNA. So it's actually more complex than is DNA. RNA is. It's not simpler, it's more complex. But the problem with RNA is its shelf life is very short. I'm talking on the order of minutes. So if, for example, you look, minutes.
Versus how much in DNA? Versus how much for, just so we feel the comparison.
DNA can be under the right conditions can be thousands of years.
Okay, so minutes versus thousands of years.
Yeah. I mean, and RNA. Now, if you take both in an aqueous system, DNA is many orders of magnitude more stable than is RNA, because RNA will self-decompose. So just by itself, the 2' hydroxyl comes around and hits the leaving group on the 3' hydroxyl and breaks the chain. Number two is the fact that you have both the 2' and the 3' can assemble. And when you hook together via the 2', it's not innocuous. It makes what's called siRNA, small interfering RNA, which slows down, which binds up to other RNAs and stops the replication process. So it's really deleterious. But this is why, for example, these vaccines, you have Moderna and Pfizer. One of them, Pfizer, was originally stored at minus 80 degrees centigrade, but now they've packaged it in a liposome or in a lipid layer to try to protect it, but still it has to be stored at 0 degrees and used very rapidly. This stuff has to be used very rapidly because the shelf life is very short. And that's only because it's packaged and there are things in there that and you have to keep it totally free of other ions. How do you do that in an environment? Our environment is full of sodium, full of magnesium. You have magnesium around. Magnesium is a big problem with these things. And so these things are unstable. So RNA is unstable. But plus, you don't have the information. Just hooking a bunch of nucleotides together and claiming that that's RNA. No, that's not RNA. That's like taking computer code and just vomiting it out there, just randomly, putting do loops here and there. I mean, it doesn't do you any good. There has to be some code behind it. So this gets into your area.
yeah, I need to stop you here because this is my next question. So it says, I just remember this quote of yours, and it's like that you make molecules for a living. So my question to you, since you make molecules for a living, and this must need so much information about what molecule and what function, how might RNA-based precursors could have acquired the needed information? And as a chemist who builds nano machines, do you see maybe there is some experiment in chemistry that is encouraging people that you would put some RNA in a test tube and, fast forward the reaction and wait until it produces a nanocar or something? Do you use trial and error to create these things like genetic algorithms, or this is, complete science fiction?
Yeah, so our RNA has a very short shelf life. So even when it's made, it's decomposing much more rapidly than the thing is made. So that's one problem. But the other problem is the ordering, the sequence of these. That's the informational code. Nobody knows where that came from. Could you have information from randomness? The answer is yes. It's called Shannon information. But Shannon information does not give you the specified information that you need to construct a cell. Shannon information grabs these pieces of randomness, but informaticians have clearly said that this Shannon-like information, which comes from information science when you're dealing with radio signals, that random type of grabbing this embedded information in randomness is not sufficient. You have to have what's called specified information in order to build cellular structure. So we don't know where the information came from. And you say, well, one randomly formed and happened to have the information. Okay, but what happens in 5 minutes when several of those pieces have been broken up? Now what? So that information is gone. And the chances of reassembling that become really, really small. So we're talking about the chances of assembling a viable RNA, something like 1 over 10 to the 40th. I mean, the numbers are so vanishingly small. So say one forms, its shelf life is very short, its half-life is very short, and then if it had the informational code, within minutes that informational code is going to be lost, never to ever form again. That's it. That's it. Even in the lifetime of the universe, a scientific lifetime of the universe, when you have 1 over 10 to the 40th, it's just too short. It's just too short. And so the problem there is information. And then even if you were to take RNA, so you have one strand of RNA, what's that going to do? Can that strand of RNA make for you then the peptide that you need to do the enzymatic assembly? You can't have RNA start building peptides by itself. It has to have exogenous other enzymes assisting it to do that. And you have to have all these other peptides available to it with the right stereochemistry. It can't be achiral. It all has to be chiral. And you can't do these chemical assemblies just like that. I mean, you have to have enzymes that help you to do this. So all of this is a real problem.
So let's say, number one, it has no objective function, so it doesn't know what it wants to create. And number two, even if it creates something, it doesn't have memory, so it loses it so fast to the extent it starts from scratch. And since it doesn't know what it's going to create, so it has no direction. So only somebody who is really not throwing long shots, but super long shots would think of this. Would you agree to this statement?
Yeah, I'd agree in the sense that there is no end game here. Molecules, as I've often said, do not care about life. They're not moving toward life. Molecules have never been known to move toward life. Organisms, living organisms, move toward life. Molecules do not move toward life. They actually move away from life. They move to a far more disordered state. And so this is not what molecules move toward. Just the simple self-assemblies that you get, the self-assemble monolayer, those are regular patterns, A, B, A, B, A, B. They're not the non-regular patterns that you need to create life. If you deal in systems, I mean, you can't build any complexity through regular patterns of A, or A, B. You have to have non-regular patterns. We have non-regular patterns within our DNA. that give us this complexity of life. You can't build this complexity just with regular patterns that you would get from a thermodynamic self-assembly.
That's great. We'll have a short break now. Welcome back, Dr. Tour. And okay, so we go ahead with this. So first, thanks a lot for the first part. We talked about general aspects of how the mainstream is addressing chance being the innovator life, and the significance of the challenge that you've posted for origin scientists to create a living cell, and why the more the time passes, the more it gets even harder. We talked about abiogenesis, how science explainers talk about it as if it's a done deal, and how sadly even some scientists talk about it as if it's a done deal. We talked about RNA oil hypothesis and how it is not reasonable to postulate that RNA just came around, assembled itself, and even with information that is meaningful, and made itself into the information-bearing molecule of life. And now we talk about something that might be very close to your heart, which is molecular machines, because for those who do not know, Dr. Tour is quite renowned in his work with molecular machines, specifically nanocars. And maybe Dr. Tour will tell us what a nanocar is, but he has he got the Honda Innovation Award for nanocars in 2005. The NSF Presidential Young Investigator Award, that was quite earlier in 1990. So he's been into innovation for quite long, and he has also in 2005 Tour's journal article, Directional Control in Thermally Driven Single Molecule Nanocars. Single molecule nanocars. So one molecule that behaves like a car, he's gonna explain this to us, was ranked the most accessed journal article by the American Chemical Society. So Dr. Tour, you know plenty about molecular machines. Can you tell us what's a nanocar, just to start with?
Sure. A nanocar is a single molecule car. We put in it four wheels and fully rotating axles. It has a chassis, and it can roll across a surface. They're so small we can park about 50,000 of them, across the diameter of a human hair, just across the
50,000.
Yeah. So you can only see this with extremely powerful microscopes, scanning tunneling microscopes.
Do they carry cargo, or they just move?
No, they can carry cargo. We have cargo carrying ones. We've made nano trains where we hook these together and make trains.
Wow.
We've made 6-wheel versions, 2-wheel versions, 3-wheel versions, so many different models we've made. And in fact, in 2017, we won the first international nanocar race, which was held in France. And we designed the car, built the car, and then it was driven by one of my colleagues from University of Graz in Austria.
How do you drive them?
You use the electric field gradient of a scanning tunneling microscope tip. And we had to go 150 nanometers around one pylon, another pylon, and then through some posts, some end posts there. And
so you need to navigate it.
Right. And we completed the race in 1.5 hours. And the next group behind us was the Swiss team, and they came in 5 hours behind us. And none of the other international teams was able to finish the race in 30 hours. So we really excelled them all in that. And then the next nanocar race, it should have been last year, but due to COVID, hopefully it'll be next year, again in France. And but we published a paper right after the race on how to build and how to race a fast nano car, because there's two things. You have to build it right, and you have to race it in the right way. And so we elevated the whole world. You know, scientists don't hide what we do. We elevate the world through that publication. So now the competition is going to be much harder because they've seen how we do it. And so it's just going to be more competitive now.
And since you've now explained it, it's even more fascinating. So I'm saying that nano cars are fascinating, but I have been, struck, fascinated by those things that God has created into us that are called motor proteins. Things like kinesins and dyneins. And for the audience, those are, those, funny robots that, like have legs and move inside our cells, and they move cargo around. So Dr. Tour, you said you needed hours to go through a racetrack of 150 nanometers. And how does that compare to the performance of a kinesin or a dynein? And I would really urge the audience, after seeing this, to go and look on the internet about what a kinesin and dynein do, and walking over those microtubules. So how does the performance of the best nanocar that you have created compare to the performance of those cars that are there in our cells?
So the kinesin, those biological ones don't really look like cars. They're much bigger than what we are. We're just a single molecule. Those kinesins are macromolecules. They're very large structures in comparison. I would say maybe more than 10,000 times larger. So we're talking at least 4 orders of magnitude larger, maybe even 5 orders of magnitude larger in length and in molecular weight as well. So they're much smaller. Our nanocars, we've built many versions that have motors on them. They're called Faringa motors. We put them in, and these motors can spin. You shine a light on them, and the motors will spin at 3 million rotations per second, and they'll move the cars much faster than we were able to move it in the races. In the races, we couldn't use a motorized car. We had to use the electric field gradient of a tip to draw it. They wouldn't let us use our motorized cars. Our motorized cars are much faster than these other cars. So with motorized cars, we could probably complete a race like that, maybe even in a matter of minutes. How do they compare? I mean, there is no comparison. Kinesin is so much more complex. Kinesin carries huge cargoes with it. Kinesin doesn't need an external individual acting on it. What it does is it moves across a microtubule, and it moves across this microtubule by sensing the electric field gradients that are going through that microtubule, and it progresses it and carries it along to bring its cargo where it needs to bring it. Those are nature's nanomachines. They're bigger than our nanomachines, but they are so much more complex, so much more functional. They do much more work than what we are able to do, far more sophisticated. And I just stand in awe of God. He does amazing things.
So, Dr. Tour, when I just saw those videos, and the kinesin it just looks like a person. So it has those legs, and it's like having its arms like this, and it's just grabbing on something, a vesicle or something, and it's like moving like this, and it's walking on those microtubules. And the walking, when I just looked into the papers that describe the chemical reactions that causes to walk, and ATP, ADP cycles, etc., and how it grabs with a microtubule, and that it has adapters. So depending on the kind of the cargo, you'll have the right kinesin, as if you have the truck that carries cement and the truck that carries glass, and it's the same truck and the same motor, but it has a different container to grab to the right cargo. And then I look into these papers that say evolution of kinesins. And what are you talking about, guys? So the question I have to you is it rational to propose that motors like this and their microtubules And it happens that ATP is around and they use it to walk, and the cargo and the adapters. Can all of this come by chance with any stretch of imagination?
Well, I think the suggestion is not that it comes by chance, but that it comes by evolution. And so
Yeah.
then you have to ask the question, give me some details on that. What do you mean by evolution? How would something like this come about? How would, tell me, and they'll say, well, one small change at a time. So how many changes would it take? How many changes would it take in DNA to make a series of enzymes to build a structure that can do that? And what's the probability of being able to build a structure? How does it know to build a structure like that? There's so many mysteries. And again, to use this term evolution, just throw it out there and to feel that that now stops all argument. It doesn't stop anything. It only just begs the question. Give me more detail. Nobody knows. Nobody knows. These things are so utterly complex. And what we're talking about, Ahmad, is just a single portion of the functionality of a cell that has thousands upon thousands of portions of functionality. And how does this one portion, how does this work? Nobody knows.
Yeah, the reason I use the word chance is that, yeah, they would say evolution and small changes, but then they say that the changes are essentially random mutation, and random is, a chance event. So from your work in motors and cars, do you put a car there and you wait for some chance to happen so that you have a better car, or maybe a car becomes a train?
No,
Is this possible at all?
That doesn't happen. Not in our lab. That doesn't happen. And it's just hard to fathom. And people will take working cells today and show their processes and somehow extrapolate this to the origin of life, and somehow extrapolate this to the functioning, the new function. So you can see, evolution is a slippery term. It's a slippery term because some people look at it in the context of making human beings. Other people look at it in the context of very small changes within a cell. There are very small changes within a cell. And if you want to call that evolution, fine. But then you have to define things for us. So what these kinesin walkers are, these are amazing new systems. We don't have examples of evolution of a new system, of a kinesin walker from something that wasn't a kinesin walker. We don't have examples of that. We don't have examples of systems changing. We have examples within a cell of small changes occurring within an organism, small changes, but never a system level change. And this is what I'm talking about.
I noticed when I traced those kinesins back that they are in eukaryotes, which is like cells like animals and plants, but they're not there in prokaryotes, so they're not in bacteria, for example. And then I need somebody to explain to me how can the whole complete system just come out of nowhere into the first, least or the last eukaryotic common ancestor or something, which is an imaginary hypothetical creature. And there is no answer. They would just say it evolved, or it, co-evolved with something. And some people would just take this as an answer. And I had a complete debate with an evolutionary biologist, and I had this as my initial thesis. And he said, I'll bring you papers about the evolution of kinesins. And it's all the same story. The paper says nothing, says it's evolution. Puts a phylogenetic tree and says it's evolution. So
yeah,
at the point it's not small changes. Yeah.
Right. I always ask people to show me the chemistry. I can understand the chemistry. Show me the chemistry on how this happens. No black box that this goes from this to this. Show me the chemistry on how that would occur. And the explanations go away. The papers don't show the chemistry on how this thing can occur, because the only thing they can say chemically is that you got a point mutation in DNA, and then you got another point mutation, and another point mutation. And how many point mutations are you going to need to change one system into another? And what's the probability of something like that happening without breaking the whole system, the original system, if you're going through all of these point mutations? That's the problem. It's really getting back to the heart of the chemistry of it.
So in your opinion, is the age of the Earth, for example, enough for some randomly duplicated strand of DNA to be mutated point by point into a kinesin? Do we have enough?
Yeah, it's not a matter of opinion. This has all been calculated. So if you take the universe, if you take the scientist number of the universe, 14 billion years, if you take the scientist number of the age of the Earth at 4 billion years, is that enough? And the calculations themselves say it's not enough to do all of this that would have to take place. So when you look at the improbability of all of these things happening, if you take those numbers that the scientists themselves give, the mathematics is going to lose. The mathematics wins over the evolutionary process. That wins all the time. The mathematics says no, evolution says yes. So that's the problem of what we're dealing with here. I don't use the mathematical argument because others have used that. Mathematicians have shown the real trouble with evolutionary suggestions. That's been shown for a long time, but for some reason they say that mathematicians just don't understand. So that's why I'm using my expertise, which is the chemistry, to say the chemistry is just not there either.
So let's go back to the chemistry. You mentioned you needed so many enzymes to create those creatures that walk all over the place in the cells. So my question to you is about enzymes and proteins now. Now, first, just for people to understand, can we have biochemistry without enzymes?
No.
We can.
No.
If you just put some biochemicals together, they wouldn't work. Yeah.
Life as we know it, no. If you want to say life is something else, fine. If you want to say a drone is life, silicon is life, fine. But as we know it, no. You've got to have enzymes. That's one of the critical components. That's like saying, can you have a car without wheels? I mean, it's a critical component.
And is it true that the majority of enzymes are proteins, and that we need the enzymes to create the proteins while the enzymes themselves are proteins? Is this a true statement?
Yes.
Yeah. So I've made this. I'm sure I'm not the first one to make it. That there is a paradox there, because you cannot produce a protein from a genetic material without an enzyme like a ribosome, and they just completely neglect that the ribosome has other things other than the ribozyme. It has many proteins there, like 80 or something, from 50 to 80 or something, and they say it's RNA, and then you can. But when you look to the papers, you find that enzymes are mainly proteins, and that they manufacture proteins. So which one comes first? How do we solve this?
Right. The RNA, people will say you have a ribozyme, so the RNA itself can be an enzyme. But we have no demonstrated example of the ribozyme functioning without associated proteins. And you need to have the RNA to make the proteins. And so it's just a very difficult problem to think about how this came about. That's why I say we're clueless.
So if the second part of the question, Mike, is speculating that RNA was doing the enzyme's role rational, given what we know today?
Well, RNA, like I said, people will say that RNA can act as an enzyme, meaning that it can be a catalyst for other reactions. But it doesn't act as a catalyst without associated enzymes. So, and even if it could act as a catalyst, it's not going to be around very long. And once it breaks, and the chances of making that again are very small. People will say that RNA duplicates itself. It's only been known in primed and human-designed RNA to be very special in its design. So it's primed and ready to go. It can only duplicate 10% of itself, generally less than 10% of itself. So that's not a duplication. So this whole idea, well, RNA, once you have it, it starts duplicating itself. No, it doesn't. It might duplicate 10% of itself in the best of cases, but that doesn't do you any good. And that 10% is not sufficiently long enough to act as a new duplication template.
Is it correct to say that when somebody just says a blunt statement like RNA can, autocatalytically replicate itself, that this is a dishonest statement?
Yeah, I don't know that they're dishonest, but they're uninformed.
Okay, but if the person who is saying that is, supposedly informed, if he is posing as an expert in the field, how would you describe such a statement? Is it deceiving to the audience?
Well, it's certainly misleading to the audience. I mean, if someone called them on it or called her on it, I would expect them to clarify that even in the best of cases, only 10% has replicated itself, and that we have no example of the RNA acting as an enzyme carrying out these sorts of functions without associated without associated proteins. I would expect them to say that.
So let's take it this one step forward, because I came to know that proteins, just if you have this peptide chain, it wouldn't be enough. They need to fold and have, this 3D structure that has the real properties, and that this protein folding process is such a big deal, and that the folding process itself involves enzymes. So I'm thinking like this: if by a stretch of imagination, I accept that one protein that is beneficial has been created through a random mutation or a series of random mutations, is it rational that we expect that even the enzymes and the other, it goes into, whatever, Golgi apparatus or something, to fold it properly, and it will find the right enzymes that will continue the process? Or if it's just a beneficial protein, it will automatically find the proper folding mechanisms. Does this come for free? Is this a free lunch? Or it needs a complete system design? As a chemist, what's your comment on that?
Well, certainly we know today that many enzymes do not fold properly into an active state without chaperones. And those chaperones are other enzymes which help them to fold into the structure they need folding. So there's a paradox there within the field of biology about how these proteins folded. And so there's a lot of study. Protein folding is a big field of study, and how you can get these proteins to fold. There are some proteins that don't particularly need chaperones, that they can go into lipids and fold properly. So some proteins need other proteins to help them fold. Other proteins don't other enzymes don't need that sort of assistance. It just depends on the ones that are there. But in order to get them to fold right, it does have to have the backbone sequence to be right. And if you get that backbone sequence wrong, I mean, things start getting mixed up. And especially things that are somewhat near the active site, or things that are key to help it to fold. So there's a real problem. It's not just the how it's going to fold. It's how you got those specific amino acids to hook together into the chain originally. Very hard to fathom how that is done. Very hard to think about how that's done without enzymes being able to hook those together. Because without enzymes, you have active side chains on many of the amino acids, and those would participate in the enzymatic coupling in ways that are deleterious to the whole structural backbone. Real problem. And we don't have answers to that. Maybe someday we will, but we just don't have it right now.
So when people talk about, all what you need is a series of point mutations, and you have all of those new protein innovations, is it correct to say that this is not a complete representation of reality, completely ignoring other things that you talked about, like those chaperones, like the folding problem, like needing a complete system really to make this thing work?
Yes, you need a complete system. So it has a point mutation, and it's going to start making enzymes from that point mutation. Then you have another point mutation, and another, and another, and another. And then you have to translate that to RNA using enzymes to translate that. So DNA translates to RNA using enzymes. And then that RNA has to translate to the amino acid sequence using enzymes. and then how it's going to get the assistance to have those enzymes fold is unknown in many cases. And so, yeah, I mean, it is a system. Anything you change over here affects what happens over here. And so many times you have to think of concerted lines of evolution. Things cannot happen in isolation. You would have to have several lines of concerted evolution happening all at the same point in time, at the same time, for something to build. And we know that even within evolutionary scenarios, those are slow events that are occurring, but now you have to have many of these coming in all at exactly the same time. It's very hard to fathom. You have to have a lot of faith to be an evolutionist. A lot of faith. I'm not sure I have that much faith.
I think they offer the antithesis of that. They say you just need to believe in science. No faith involved.
It's
right.
faith. It's faith at every step.
It's faith at every step.
Yeah.
Except that there's nothing to have faith about, because this is nothing. So anyway, so I'm just worried about time. I wanted to ask you about cell membranes and the proteins there, but I will maybe we'll have another discussion about that. But let me ask you this: some are giving up on the RNA world hypothesis. So this one was about if cell membranes needed proteins, and where would those come from? And since you need them for the membrane, then you need the proteins in the membrane, and the proteins need the DNA, and then you need the enzymes, and then if you don't have the membrane at the beginning, you don't have a cell. So it's a paradox. So in a nutshell, would you agree that this is a paradox on its own, that you need proteins in the membrane and you cannot have a cell without the membrane? And
absolutely. So these things of protocells, where they just have a lipid bilayer, which you make by applying shear forces to a lamella. It's not how soap works. Soap does not have a bilayer membrane. Soap has the hydrophilic things on the outside, hydrophobic ones on the inside. The lipid bilayers have hydrophilic, water-loving things both on the inside and the outside. So you have a lipid bilayer. Some people get confused between what a cell has in a lipid bilayer and what soap does to have a monolayer where you have polar on one side, non-polar on the other. But in any case, to have this, it can't just be that you can't just be the lipid bilayer. You have to allow certain things in and certain things out. So you have to have proton gradients. You have to allow certain things in, certain protons out, and this is all controlled by proteins that are opening and closing and controlling all of this. These are highly complex structures, and the other thing is you need enzymes to make the lipid bilayer, because lipid bilayers cannot be symmetrical. They have to be non-symmetric, meaning the outside has to be different than the inside. If you just take lamella and put them under shear, you get some symmetry between the outside and the inside. That would not be suitable for a cell. You have to have asymmetry across this. And the asymmetry right now we know is induced biologically by enzymes called flipase enzymes that flip certain things to the outside and certain ones to the inside.
It just gets more complex as you go, doesn't it?
Yeah. So where are you going to get your lipid bilayer if you don't have the enzymes to do the flipping for you? It's a real problem. And people will say, well, they were much simpler in the beginning and those things just evolved. No, that cannot have been, because you would not have been able to have the proton gradient to have your simple cell. So you cannot keep throwing this right at the feet of evolution and say evolution took care of this, or evolution becomes your God. You throw it at the feet of evolution, evolution took care of it. Well, how is that any different than just making evolution then your God?
This is actually the next question. So I just pursuing this RNA world hypothesis, and it seems that some people are, letting go of it, and they say it wasn't RNA alone, but peptides and RNA just evolved side by side, and it's a love story. And they have those drawings. It's not mainstream yet, of course, but does this solve it?
No, because because hydrolysis of peptides means the breaking up of peptides is faster than the formation of peptides when you don't have enzymes putting them together. And chemists will say, oh, we do it all the time without enzymes. Yes, because you activated the end of one and you had the other thing protected, and you set it up to do that, and you protected the side chain. How do you do that when you don't have a chemist acting upon it? Nobody knows. Nobody knows. So
if you don't have a chemist and you don't have an enzyme, you're hopeless. Is this what you're saying?
Yeah. I mean, it's really hard to do this. And so the hydrolysis rate is going to be faster than the formation rate. And chemists that say, oh, well, these things can happen, we'll dissect their chemistry. This is going to happen. We'll dissect the actual chemistry that they did, and we'll take a look at it. And the nice thing about a real chemist is that when you point something out to them, they'll concede. They'll concede. I mean, because you just can't argue against what's there. And so even the real chemists that are picking on me, it's for very tangential things. They haven't gotten to the heart of anything that I said in that video. Because if they listen to that video, they're going to go, wow, he was right. He was right, over and over again. And those that have contested it will expose it.
So, Dr. Tour, your series was about origin of life. Let's talk about what is life. And you, I have, it's not for me to give you the credit, but I have to give you the credit that you are the first one who let me hear about interconnectomes. All right. And I've been reading about this thing for 20 years, and the first time I heard about the criticality of the matter of interconnectomes was from you. So what is an interconnectome, and why is there silence about it in the world of microbiology, to the extent that I don't think it's my ignorance. I think it's very rarely talked about.
Yeah,
why is there silence about it?
Well, it's not that there was silence in the sense that these are newly discovered things. This is what I'm telling you. The goalposts move further and further away. We're just learning about, and this interconnectedness is, I think the term is interactomes. And so these. My bad. Yeah, that's all right. And so these interactomes, this is the non-covalent interaction that occurs between molecules. So you can have molecules hooked together. So they're hooked together by a bond. That's a covalent interaction. Or you can just have them associated with one another because of non-covalent bonds. And you might think of it analogous to, it's not this, but it's analogous to magnetic forces. So magnetic forces that it may not be as strong as hooking things together physically, but there's reasonable strength. That sort of force between them. It's this interactome force, and it's what's called all the forces put together are what's called the van der Waals forces. All of these forces put together, and it turns out this alignment between them is critical for life. And so you have extraordinarily high numbers of possible arrangements when you're talking about interactomes. So the one I gave, the one that's used in the article, which I talked about in my video, is that there are the chances of getting the proper interactome that you would need just in a single yeast cell, which is a very simple cell, just thinking of the protein-protein interactions, not protein, DNA, protein, RNA, just protein-protein, is 1 in 10 to the 79 billion. This number is so big that it's crazy to even think about it. 1 in 10 to the 79 billion. So the numbers.
The first time I heard you say that, I thought you made a mistake.
Right.
Let's just be sure. It's 10, and there in the power there is a 1 having 79 billion zeros in front of this, and this is in the power.
Yeah, 10, and then it's 79 0 0 0 0 0 0 0 0 0.
Yeah. 79 billion.
Yes, 10
in the power.
In the power, right.
Okay. Not that we have 79 billion interactomes. No, it's
Right.
10 to the power 79 billion.
Billion possible
This
combinations.
is an unimaginable number, essentially. Nobody can imagine it.
It's unimaginable number. And this is not my number. And that's why I showed in the video, I showed the actual, here's the quote from the paper. It shows it, 10 to the 79 billion. And so you have these types of numbers. And so now you have a new paradox that's not just the protein folding paradox and the protein coding paradox, where those are, in the 1 in 10 to the 40th or 1 in 10 to the 50th. This is a much bigger number. So what happens, what the cell does, life is remarkable. So what a cell does is it takes this informational arrangement, and the cell, when it divides, it clamps down and it takes half of it here, and now you get 2 new cells. So it keeps passing the information along.
Can I ask you, how do we know that the interactomes have informational content? So how do we verify the hypothesis?
Right. So it has informational content, because if you lose it, the cell cannot function. All right, the cell stops functioning. So when you lose structural water, these are held together by hydrogen bonds, for example. You lose structural water, you can never rehydrate a cell once you've lost the structural water to a cell.
So this is a testable hypothesis.
This is testable, and the other thing about it is, through these non-covalent interactions, that electrostatic potentials transfer. Electrostatic potentials are these electron gradients that pass information through cells. Information is transferred through electron gradients. And the reason we know things about this is, you can have, say, a DNA strand, and we suspected this. Now, a DNA strand is covalently linked, but the idea was, how is this an enzyme here on DNA, hundreds of angstroms away, know that there's a break in the DNA or damage down there, such that the enzyme starts moving down there? So it gives us clues that there are electron gradients that are going through the orbital cloud system. You can show, and we've published in this area, we've published papers in this area to show that non-covalent interactions have a very long coherence length of information transfer, electrostatic information transfer, that goes through the orbital construct, such that you affect one orbital, it affects the next, and this travels at the speed of light. This is much faster than ionic gradients. This is at the speed of light, and near the speed of light, these electron gradients, these transfer gradients. And all of this is information content. And so this is giving us clues that within a cell, there is this way of transferring information that's just remarkable. This is the complexity of a cell.
So when I'm watching a video about damage to a DNA strand, and then this enzyme comes and just lands in the right place and starts fixing it, it's not because it has randomly gone there, or it is continuously, policing the whole DNA or the whole chromatin or whatever, or the whole chromosome. It is because it received a signal.
It receives a signal. So it could be way, way far away and just start moving right across that DNA to fix that break. If it had to randomly find mistakes on DNA, we'd have a whole lot more cancer than we have. And we'd have a whole lot more problems. It is getting electronic information that is signaling it to do this. This is extraordinary. And what happens is, I've seen people even make videos and say, well, Tur grabs these esoteric concepts like chiral-induced spin selectivity. And so because they say it's esoteric, it now doesn't have to be addressed. No, this has to be addressed. This is what I'm talking about. Most biologists have never even heard of interactomes. Most biologists have never even heard of chiral-induced spin selectivity. What I'm bringing to light here is the complexity of the chemistry of the new things we're learning before the biologists even start talking about it. And YouTubers will say, oh, well, that's just esoteric, so somehow it doesn't have to be addressed. No, it has to be addressed. This is what I'm saying. The complexity is getting harder all the time.
Yeah, and I have always wondered, when I'm watching those, rendered graphics of, an RNA strand getting out of the nucleus and miraculously it finds, the ribosomes, and miraculously then the protein finds another enzyme to fold it, and then miraculously it finds a kinesin or something to move it around. What you're saying, that this is a system that is signaling itself all the time to find itself and act, and that water has a critical role there, and the arrangement of all those van der Waals interactions is not random. This is a system that is looking for a chance to be properly arranged in 10 to the power 79 billion possible ones, and not all of them will make it work. A very few of them will make it work.
Yes. This is the complexity of a cell. That's why I said your fly is just, you can't even think about that. I mean, it's over. The argument's over if you say a fly.
All right, so it just brings us to the water, because you talked about the water. So interactomes related to the matter of life being dependent on water, and I think in all religions, water has this, elevated status. And what you're teaching us now is that water is just not those, passive H2O molecules. They are a communication system. They are an information store. They are memory. They are much more than what we thought.
Yeah, it's glue for the interactomes. I mean, these hydration pathways are essential. If you just take the water molecule and you just change the dipole moment of water, if it were just 0.001% different, the dipole, no life exists. I mean, this is the whole argument of fine tuning. Everything has to match up for this. Just has to match up for this. I mean, water's amazing, and it has to have everything has to be right for this to work. I think I'm going to have to move on here, and we're going to have to wrap this thing up.
Let's go into closing, and this was super exciting, Jim, really, and I wonder if you'd like to continue this some other time, but let me jump to I have some few closing questions. I'll just wrap them into one. I had one about God, and about how it feels to be a believer in science. And let me ask you this one. Can you tell us about one thing in your work where you saw God's hands so obvious? You thought, this can never be there if there is no God.
Yeah, I remember we were building what we had called a synthetic brain. And it was to take a disordered array of molecules and nanoparticles in a box, in a micron-sized 2D square, and program it to do something useful. So to take disorder and give voltage pulses from the outside to make order. This was
the constituents. What were the constituents?
It was molecules that could undergo switching states and held together by nanoparticles. And we were able to make very, very simple little things like an AND gate, like an OR gate, like a NOR gate.
So those are like biological transistors, let's say, more or less.
Something like that. But then at the same time, my son, who was about 4 or 5 years old at the time, came running toward me. And I looked at the complexity of what he was saying and what he was doing and how he's running and moving. And here I am putting everything I have into a molecular system to try to build some sort of computing device. And here is a functioning child. And I said, God, you are extraordinary. How did you do this? How did you make him like this? This is just utterly amazing. And this is the beauty of being a scientist. You get to look at the things that are ubiquitous around us, and you say, this is utterly amazing. How? And I just had to give glory to God. I had to give glory to God. I love God so much. He means everything to me. I love serving him. I love honoring him. I just enjoy doing my work and knowing him. And when I look at biology, And then I look at the simple things that I do. I just give glory to God. I said, Lord, you are extraordinary. Blessed be the name of the Lord. Blessed be the Lord Most High.
I think that was too marvelous to say anything after it. It's just consistent with what we say, that real scientists are the ones who glorify God. Dr. Tour, I don't know. I'm endlessly thankful to you for this amazing talk, and for sure, I'd love if you can spend some more time here. We had some questions from the audience, but most of the comments are actually commending what you were saying and agreeing to it. So thank you so much for being here.
Thank you.
It's a blessing to find somebody who is as excellent as you in your field, and as loving and touching in your feelings towards the Creator. And there is one God for everybody, and I thank you for seeing this, and for really thank you so much for having all the integrity and the courage and the honesty to call this out, and be the one. Everybody's looking at you and up at you now, because stand strong there, Dr. Tour, and thank you very much for being here with me today.
Thank you.
Thank you so much. So everyone, thank you for watching this, and God bless Dr. Tour and your pursuit of truth and pursuit of science and service to humanity, through the great things that you're doing.
Thank you.
And see you again soon.
Bye-bye.
Hopefully. Bye-bye.